fMRI. MRI can be used to detect the presence of different molecules like water and fat because their magnetic properties differ. Turns out oxygenated blood and deoxygenated blood also differ in these properties. Therefore, if we can set up the MRI scanner to focus on this contrast between oxygenated blood and deoxygenated blood and take a picture every few seconds, we can create a movie of how oxygen is used in different parts of the brain over time. And that is fMRI. It's called fMRI because it uses MRI to measure and assess brain functions. This is possible because when neurons fire, i.e. when they send out a signal, they use up some oxygen in the blood. You may think that the more the neuronal activity, the bigger decrease in oxygen there is, but in reality, the initial decrease in oxygen is compensated by a subsequent influx of oxygenated blood, and it is this delayed blushing effect that is more noticeable. So in fMRI, we measure the increase, not decrease, and in the ratio of oxygenated to deoxygenated blood a few seconds following neuronal firing. We call it the blood oxygenation level dependent signal, or BOLD. So this BOLD signal, as you can see, is a very indirect measure of neural activity. It does not track the activity specifically in meaningful units like spikes per second. The relationship between specific firing to the change of blood oxygenation is complicated. So often the BOLD signal is just expressed as percentage change from baseline because it is difficult to give it a more precise and meaningful unit. The BOLD signal changes on the order of just 1% or 2% change, sometimes smaller. Also, the signal is sluggish. It peaks at about 5 to 6 seconds after the initial neuronal firing. This time course is sometimes called the hemodynamic response function, HRF. The good thing about fMRI is the spatial resolution is pretty good. If we push for the highest resolution, it can be smaller than a millimeter. That is, each pixel or voxel, because it is in 3D, can be smaller than a single millimeter cubed. Though usually, for most purposes, we keep the voxels at about 3 millimeters cubed or so. So usually, in an fMRI study, we put people inside the scanner and get them to do various psychological tasks like watching a movie or counting numbers backwards. Because the BOLD signal is sluggish, one easy way is to get people to do one task for 30 seconds or so, and then rest or do another task for 30 seconds. This is called a block design of the experiment. But sometimes we can switch between the task conditions faster, say every few seconds. That would be called an event-related sign, in which we have to mathematically sort out the delayed sluggish signal which may overlap each other. Sometimes we say we deconvolve such signals, so we know how much activity is caused by each event. So fMRI measures the BOLD signal over time for each voxel. How come in papers and news articles we see pretty pictures like these? Typically, these color scales reflect the statistical score for comparison, meaning how consistently one psychological condition gives bigger BOLD signal than the other. Usually, this consistency is measured across subjects. Essentially, we repeat this procedure over every voxel and color code the statistical score for each contrast of comparison. When this score is higher than the threshold, we apply color to the voxel. So it is important to realize the brain region never lights up itself.
What?
When it does, it just means that our statistical measure has reached a certain level of significance. Also, when an area doesn't light up, it does not mean that the two concerned task conditions give exactly the same level of BOLD signal. It just means that the difference is not consistent enough. Maybe the difference is small, or maybe the data just varies across a lot of participants.